So hello and welcome to another class of ABME Biosciences. This is Abhishek with you. So today I'll be talking about the last stage of the protein synthesis, that is termination, and also I'll be talking about the protein termination recycling process. Means after termination, how the process is again recycled to take another synthesis or another polycistronic mRNA protein synthesis. So here let's start with the termination. Now, last day we have talked about the elongation, where you have seen that the P site is continuously synthesizing and it is transferring the amino acid tRNAs with the polypeptide, and it is transferred to the next tRNA, and the polypeptide is transferred to the with the tRNA is transferred to the P site with the translocation process, that is EF-G. Now here, to terminate the process, the cell when requires it to terminate, that time it release or it requires or it depends on some of the processes. Like in termination, there are 3 termination codons that you already know, 3 release factors which are very essential in this process of termination. Now the release factors are RF1, RF2, RF3. So RF1 recognizes, why it is different, I will tell you the functions individually. Actually it recognizes UAG and UAA, whereas RF2 recognizes UGA and UAA. Now what is the function of RF3? I will tell you, just listen to the class. You will be listening to the function of RF3 in between, but I will not say to now itself, it will be confusing for you. So as the picture shows here, you have seen this is the tRNA bound with the polypeptide chain of the proteins in the red color. Here you have seen the termination codon UAG recognized by the factor, that is RF1, and this is 50th, 30th subunit including mRNA. Now, when a ribosome encounters a termination codon, here it is UAG, that time release factors like RF1 and RF2 will bind to the codon, that is termination codon, and what they do, they will just transfer the polypeptide. The normal rule is to transfer the polypeptide to the next tRNA, okay, and form the peptide bond. So they will induce the 23S rRNA, induce 23S rRNA to transfer the polypeptide, to transfer the PP polypeptide to H2O, that is a water molecule. So the ester linkage is broken. Now the proteins which needs to be transferred to the next tRNA, that is not transferred to the tRNA but instead of a water molecule. So what happened in the next step? That is dissociation of the polypeptide from the tRNA. So here if I draw this, so this polypeptide is now detached. So detached from the tRNA which is present in the P site. So up to this, that RF1 or maybe RF2 if there is a UGA. So that time RF1 and RF2 it will be competing with each other, and 1 or 2 will be binding there, and it will release the polypeptide from the tRNA. It is a step of the termination process, but still there is a P site where the tRNA is bound and the tRNA is deacylated, means no polypeptide or amino acid is bound, and one A site is there where release factor RF1 or RF2 is bound. Now here comes the RF3. So what RF3 will do, it will release the RF1 or RF2 from its position. So RF3 induces release of RF1 or RF2 from the complex, that is, I will say here, post-termination complex. Okay, so now you know the function of RF1, RF2 and RF3. So now if you just compare the scenario in your mind, I'll just wipe it down for your ease of understanding little bit. So now here so when the post-termination is formed, PTC, post-termination complex is there. There is the A site which is empty now, and P site where the tRNA is bound, and there is E site and there is a 70S complex. The total thing is 70X complex. Now sometimes another factor, RRF, that is ribosomal recycling factor, ribosomal recycling factor comes into scenario. So what happens that RRF, which is mimicking to a tRNA. So remember that thing, RRF mimics like a tRNA, which will bind now to the A site. Okay, by competing what? If there is RF3 is bound, it will compete with the RF3 and it will release the IF3 and bound with the A site like RRF. So now RRF is bound to the A site, releasing the RF3. Now this RRF, after binding as a tRNA analog to the T site, here comes another one, that is, you already know, EF-G GDP. So EF-G you have studied in the elongation process, which helps in the translocation of the process. Now here EF-G is again in the scenario where EF-G will come and bind with the or interact with the RRF with the help of GTP exchange, and it will form a RRF EF-G GTP. So EF-G is coming as a GTP. Now it transfer in another form, that is GTP, and interacts with the RRF, and it induces a translocation or mechanical change to the post-transcriptional complex, that is 70S, which is now transform itself, or you can say translocate from one side, such a translocation that P site, this tRNA will now go to E site and it is released, whereas this RRF with EF-G is now in the P site, and there is a transient dislocation of the total 70S complex, and this time only this 30S complex is bound with IF3. Yes, you are listening correct. This is IF3, which you have studied in the reinitiation process. This is the initiation factor A3. Now the EF3 is bound with the 30S subunit, and this 30S, after binding to 30S, the 50S will not be able to bind with the 30S and will not be able to form the 70S ribosomal complex or maintain the 70S ribosomal complex structure. So it will now dissociating. So the 30S, suppose this is the 30S is dissociated, this is the 50S is dissociated, the RRF and EF-G is dissociated, the mRNA is separated, and the polypeptide was previously separated at the time of release factor binding. So you can observe that how the termination is not only the termination, but also it is creating a recycling again of the same ribosomal complex with the other factors, that is the initiation factor IF3 already bound. So now the 30S ribosome is again ready to bind with another stretch of mRNA and start the protein synthesis as soon as possible. So again IF1 or IF2 will come, and it will bond with the, it will help in the binding with the 50S and 30S ribosomal subunit from the 70S, and again the elongation process will continue. So this kind of termination and release recycling is what can say accompanied process. So at a time like in the prokaryotes, like transcription and translation is simultaneously happening. That time also after termination, recycling is also simultaneously happening and maintaining the 30S and 50S subunit, or inducing the 30S subunit and the 50S subunit to form as soon as possible the correct structure with the correct formation and binding with initiation factors or the release factors to form another complex with the next mRNA. So I hope this is not so confusing. So if you have any doubt, please ask me with mail and comments, and do subscribe or like my channel if you like my classes. Thank you. So if you like my classes, give a big thumbs up and don't forget to subscribe my channel, because these things stimulate me to take futuristic classes for you guys. So thank you and be with me.